hello students. Today we'll learn about antigen processing and presentation, which is a critical mechanism that elicits a successful immune response. Right? So as we all know that antigen processing and presentation is one of the key mechanisms to elicit an immune response because by definition, you know what is an antigen, right? So an antigen, it could be a virus, or bacteria, or any other microorganism, right? Foreign material that enters into the body, right? So our body, as it is, it cannot capture the whole microorganism. So it has to process the particular antigenic portion of that particular antigen, right? So for example, we learn about COVID virus, right? So SARS-CoV virus. In that, the spike protein plays an important role in the pathogenesis, right? So in this case, the whole virus cannot be processed or captured by our immune system. Only a part of antigenic portion, that is spike protein, is captured by our immune cells to elicit a successful immune response. So let's take a quick look. What are those cells that process an antigen and aids the presentation of peptide to major histocompatibility complex molecules, right? So antigen presenting cells are basically divided into two different types. One is professional and non-professional antigen processing cells. So professional antigen processing cells, they include dendritic cells, macrophages, and B cells, whereas the non-professional includes fibroblasts, glial cells, pancreatic beta cells. Right? So therefore, any cell that can process and present antigenic peptides in association with either class 1 or class 2 MHC molecules and deliver a co-stimulated signal necessary for T cell activation. Right. So this pathway depicts the pathways, two different pathways involved in antigen processing presentation. That is exogenous pathway and endogenous pathway. Right? So exogenous pathway is also known as a cytosolic pathway, in which the antigens are generated within the cell and peptides are generated by the system known as proteasomal degradation system. We know that system aids to degrade proteins into smaller peptide molecules. So this exogenous pathway specifically presents the processed antigen to class 1 MHC, and that class 1 MHC has a predilection to a specific T cell subtype known as T cytotoxic cell, right? So on the other hand, the exogenous pathway, the peptides are presented to a class 2 MHC molecules, which has predilection to another subtype of T cell known as T helper cell. So in a nutshell, the class 1 MHC molecule presents a processed antigen or peptide to T cytotoxic cells, whereas the class 2 MHC molecules, they present a processed peptide to T helper cells. So let's take a look how these peptides are generated. What is the underlying mechanism in exogenous pathway? Right. So this is the overview of both exogenous and endogenous pathways that process the antigens into smaller peptides. The left panel shows the endogenous pathway in which the class 1 MHC plays a key role. So this class 1 MHC, it captures an antigen as a whole, and this antigen is then processed by a mechanism called proteasomal degradation system. It takes the help of proteasomal degradation system, and then once it is processed, it will be delivered triggered by this Golgi complex, and this will present, or this will protrude out of the cell to present this process peptide to class 1 MSC molecules. Right. So this is the proteasomal degradation system. In general, what happens when there is accumulation of proteins, or there is a misfolded or abnormal proteins that accumulate within the cells. So they have to efficiently eliminate by the cell, right? So what happens? There is a molecule called ubiquitin that attaches to these abnormal proteins. So that's a tag. So when ubiquitin attaches to this abnormal protein, the proteasomal degradation system recognizes those abnormal proteins, and as a result, it will degrade those proteins into smaller peptides. So this mechanism will be employed by this endogenous pathway to process peptides. So in addition, the endogenous pathway also takes help of a protein called transporter associated with antigen processing. So this particular protein, it's a peptide, and it has an affinity to a peptide that contains 8 to 16 amino acids. So this is the optimal peptide length for accommodating a peptide into the class 1 MHC peptide binding group. That means class 1 MHC peptide binding group can only accommodate about 9 amino acids. So the tag protein, it helps to trim the longer amino acids. For example, if there is 8 to 16 amino acids, it will trim down to 9 amino acids so that it can facilitate the binding of this processed peptide to class 1 MHC. Right? So this is the overview of where the processed antigen is bound to the class 1 MHC molecule and exits the ER to present to the T cytotoxic cell. Right? So another pathway is exogenous pathway. Right? So now we learn about endogenous pathway. And what is exogenous pathway? Exogenous pathway means the antigens coming from extracellular region. So they enters into the intracellular region by a mechanism called endocytosis, right? So once they enter, as I earlier mentioned that this exogenous pathway presents a processed peptide to class 2 MHC. So class 2 MHC has predilection to T helper cells. So in this case, class 2 MHC is apparently synthesized in the endoplasmic reticulum, because endoplasmic reticulum is the site of protein synthesis where all proteins are synthesized. Right? So here in this case, the class 2 MHC molecule is synthesized within the endoplasmic reticulum, and that also contains a specific unique chain known as invariant chain that binds to the peptide binding group of class 2 MHC molecules to avoid binding of any unwanted proteins during its translocation from endoplasmic reticulum to the Golgi complex. Right? So this invariant chain remains within the MHC group until it binds to the exogenous antigen. Right? So once the exogenous antigen is present, binds to this class 2 MHC molecule, so this invariant chain dissociates, and the processed antigen that binds to this peptide binding group, and that will be presented to T helper cell. Right? So T helper cell, it helps to activate B cells. Right? So B cells, B lymphocytes, are the cells that are important, that are critical for the production of immunoglobulins, right? Antibodies. So that is how our immune system works. And this T helper cell, it differentiates into two different types of cells. One is a memory cell, right? One is effector cell. So memory cell, it will retain the memory for the same antigen. So whenever we encounter with the same antigen for the next time in the future, so it can quickly elicit an immune response. right? So on the other hand, T cytotoxic cell, it just kills the cells without aid of any antibodies, right? So the vaccination process, now we know the vaccines. Nowadays we use vaccines, many vaccines, including recently we all took shots of COVID vaccines. So the underlying principle is this, the class 2 MSC mediated presentation of antigen to T helper cells, and subsequently that activates a B cell to produce a memory cell and specific antibody to degrade the pathogenic bacteria or virus. I hope you understood well, and I'm happy to answer if you have any questions regarding this. Thanks. Any questions?